Thank you. All right, everyone, thank you so much. Welcome to our Emerging Clinical Trials for New Therapies session featuring Neuren Pharmaceuticals and Jaguar Gene Therapy. Understandably so, this is likely the most, if not one of the most anticipated topics at the conference in general. It's important to note that while there's so much exciting work going on, clinical trials are still experiments. I felt as the foundation that we need to say that. They are designed to be a test for a treatment and people for safety and effectiveness, as Dr. Barry Kravis just explained. And participating in a trial is a really personal decision. If you visit our clinical trials page, we have a rubric for what to consider before joining a trial, and Dr. Barry Kravis covered a lot of that. With that being said, please enjoy this session focused on emerging trials. We are happy to have two industry representatives with us today. First, we have Dr. Liza Squires, Chief Medical Officer at Neuren Pharmaceuticals. Liza's career has focused on the development of novel therapeutics for diseases of the central nervous system that affect children. Prior to her current role, she has 20 years of experience and has held positions in increasing responsibility at both early and late stage companies, and including Johnson & Johnson, Shire Pharmaceuticals, Lumos Pharma, and Origin Biosciences. And next, we have Dr. Dan Gallo, who joined Jaguar Gene Therapy in January of 2022, where he leads clinical development strategy and medical affairs. During his tenure with Jaguar, Dan has worked closely with academic partners, advocates, and collaborators, including PMSF, the Developmental Synaptopathies Consortium, and CureShank, to develop and advance Jaguar's clinical program for JAG201 and SHANK3 haploinsufficiency. Dan has a PhD in cell and molecular biology from Northwestern University and has over 12 years of experience leading and supporting drug development in the rare disease space. I think you've probably seen both of them at various webinars that we've done, and they will both cover updates from their current drug development programs, and then we will leave room for questions. So first we will do Dr. Squires' presentation and questions for her, and then follow with Dan's presentations and question for him. Thank you all.
Good afternoon, everyone. It's just a pleasure to be here, and I'd like to congratulate the PMSF and all the volunteers for putting together an amazing meeting. It's our team is here, and we're just really happy to see and meet everyone and to collaborate with our partners. Today, I'm going to talk about NNZ-2591 for the treatment of Phelan-McDermid syndrome and share some of our results from the phase 2 study. Dr. Barry Kravis did a great job. She really stole my thunder on what exactly NNZ-2591 does. And the long story short is it regulates the amount of IGF-1 that's able to interact with the receptors, and therefore activating really specific signaling pathways which regulate the formation of new synapses. The few things that maybe I'll just add or stress is that NNZ-2591 is really state dependent, and that it appears to correct impaired cells, but it doesn't have an impact on cells that have normal function. So here's the study design that we're going to present to you today. The objective of the study, as Dr. Barry Kravis said, it was a phase 2, so we're really looking at safety and we're exploring efficacy. And one of the major goals of the study is to get information on efficacy endpoints that we can bring into the phase 3 or pivotal trial. The study enrolled 18 children and an adolescent who happened to have their birthday during the trial and turned into a teenager. And it was conducted at 4 sites in the United States. The primary endpoint, as I mentioned, is safety, tolerability, and pharmacokinetics. And secondary endpoints were efficacy measures, 5 of which were PMS-specific endpoints, including global and symptom-specific endpoints to PMS, and then there were other endpoints that are more widely used across a variety of pediatric conditions or neurodevelopmental disabilities. The study design, which is in the middle of the slide, shows that following a baseline observation, which was 4 weeks, and this really gives us a chance to understand the ebbs and flows of certain symptoms. Then at week 4 is the baseline visit, and this is when the child receives the first dose of NNZ-2591. Treatment was given twice daily for 13 weeks, and the first 6 weeks were dose escalation, and children dosed from 4 milligrams twice a day to 8 and ultimately to 12. And then there were 7 weeks of a dose stabilization period, and then after ending treatment at week 17, there's a 2-week safety follow-up. So I'm going to talk a little bit about the PMS-specific measures. They're including, and many of you may have heard of these before, the Clinical Global Impression of Improvement, and in a similar vein, there's the Caregiver Impression of Change. Now, these scales are rated from 1, which is very much improved, to 7, which is very much worse. And the only difference between the CGI-I and the CIC with respect to scoring is that for the Caregiver Impression of Change, instead of 3 and 5 being minimally improved or minimally worse, we just leave them at improved or worse just to aid in scoring. There's also the Clinical Global Impression of Severity, and this reflects the severity of the disease as rated by the clinician on a 7-point scale, from 1 being essentially normal to 7 being extremely impaired. The clinician raters undergo comprehensive training and calibration of scoring to anchor the interpretation so that a CGI-I of 2 in Boston is the same essentially as a CGI-I of 2 in Chicago. So the domains of the Clinical Global Impression of either improvement or severity, the scoring is done with the clinician giving an overall impression score as well as rating specific domains. And these domains are specific to PMS and come out of a lot of work that the foundation participated in to identify the symptoms that best characterize PMS. So we talk about expressive communication, receptive communication, gross motor function, fine motor function, social interaction, cognition, learning, and self-care. For the caregiver impression of change, these domains are very similar, but lightened up a little bit, and there are a few extra domains that we look at. So communication is just simplified to communication instead of expressive or receptive. Motor abilities are just graded overall, which would be inclusive of both fine and gross motor. Social interaction, cognitive abilities, abilities to learn, self-care. But then additionally, we had the caregivers rate behavior, GI problems, and sensory sensitivities. There were 23 children screened for this study. 5 did not meet eligibility criteria, and 18 received drug. 15 children completed the study, and 3 discontinued, none of which were considered to be related to study drug by the investigator. The mean age of the participants was 8.6 years, and the range was from 4.4 to 13. There was a predominance of males in the study, with 67%, 33% female. This isn't due to any background prevalence difference within the disease, but is probably just the result of a relatively small sample size. The Clinical Global Impression of Severity at baseline had a mean and a median of 4.5, which is between moderately and markedly ill. And NZ2591 was generally safe and well tolerated in the study. Most of the adverse events that occurred during the study were mild to moderate. There was one serious adverse event of gastroenteritis not related to study drug, and this actually occurred in the safety follow-up period, that 2 weeks from completing drug to having your final safety visit. 3 discontinuations due to adverse events. Again, none were considered related to study drug. 2 were due to testing positive for COVID, a sign of the times, and 1 was due to seizure disorder. There were no meaningful changes in laboratory values, ECG findings, or other safety parameters observed during the treatment period. On the right-hand side of the screen, you'll see the adverse event table with the most common adverse events leading and going downward to the least common adverse events that occurred in 2 or more participants. Most common adverse event was psychomotor hyperactivity occurring in 4 subjects, COVID-19 in 3, and decreased appetite and pyrexia in 3. 3 subjects had somnolence, which is really drowsiness, and 2 subjects had aggression, and 2 patients had constipation, and 2 had diarrhea. So to summarize the efficacy endpoints, 10 out of the 14 efficacy endpoints showed significant improvement when compared to baseline. The PMS-specific measures, the global measures, the clinical global impression of improvement, as well as the clinical global impression of severity and the caregiver impression of change were all statistically significant. The symptom-specific endpoints, including the PMS domain-specific rating scale, which looked at PMS symptoms that may be are characteristic of the disease, but not as widely appreciated in each and every patient with PMS, such as GI problems, seizures, and the like. In that scale, as well as the caregiver top 3 concerns, they were all statistically significant as well, and we'll go over those in some detail later in the presentation. The other endpoints that were notable included the GI health questionnaire, the child sleep habits questionnaire. For quality of life, we looked at the quality of life disability overall score, which is a single 6-point rating, and the ICND is the 6-point rating. We did not separate on that, but we did separate on the quality of life disability scale. We also looked at behavior scales, the aberrant behavior checklist, and the behavior problems inventory. We did not reach statistical significance in the communication domains of the MacArthur-Bates or the ORCA. Came close. This may have been a function of either a small sample size or perhaps a shorter study, 13 weeks. Also, we looked at the Vineland adaptive behavior scale and did not separate on that. Again, could have been sample size or the short time frame. So just a little more detail about the PMS symptoms as measured by the clinicians in the CGI-I. 16 of the 18 participants showed improvement by the end of treatment. On the left-hand side, you'll see what we call a bar chart. On the up and down, or the y-axis, the top is very much improved, the best score you can get, and at the bottom is 7 and very much worse. And each of these bars represents a participant in the trial. So you can see that 2 patients were rated by the clinician as very much improved, 8 as much improved, 5 as minimally improved, and 2 as unchanged. On the right-hand side of the screen is the specific domains of improvement that were rated. And you can see that the further you're away from the number 4 with the vertical bar, the more improvement, because you're looking at the one on the far left-hand side, and the 7 is on the far right-hand side. And you'll see a recurring theme that there's the domains that seem to improve the most were expressive communication, cognition and learning, receptive communication. So we're really looking at this kind of communication and social interaction domains showing improvement, and as well as cognition and learning. Now, all of these endpoints did reach statistical significance, including gross fine motor and self-care. We look at the caregiver rating. 15 of the 18 participants showed improvement at the end of treatment. 2 patients were rated as very much improved, 5 as much improved, 8 as improved, 2 as unchanged, and 1 subject got worse during the clinical trial. Again, on the right-hand side, you see the specific domains that were rated by the caregivers, and statistical significance was reached on the communication, cognition of learning, and social interaction and behavior. Because of the small sample size, sort of a poor man's way to look at statistical significance is to go straight up from the unchanged bar that's vertical in that plot. And if everything is to the left of that number, often when looking at these kind of charts, it may be statistically significant. However, we're dealing with a smaller sample size here of 18 subjects. So now we have the PMS severity. So this is really how ill overall the participant in the trial is as judged by the clinician. This is a much higher bar to move than improvement, right? We can see incremental improvements. This is really more of a monumental improvement to even move by 1 point into a new disease category or a new rating category. And in this trial, 7 out of the 18 participants did improve by 1 point on the CGI-S by the end of treatment. The areas, again, that clinicians saw to be most improved included cognition and learning, social interaction, and fine motor function. The caregiver top 3 concerns is a analysis where we ask the caregivers to talk about the top 3 things that they would like to really see improved for their child. And in this case, you can see that on the y-axis, the various concerns that are rated and the number of caretakers that nominated those. So the most common ones, you can see communication is 13, behavior 10, social interaction is 7, and then there's, self-care was also 7. And statistical significance for these was reached in communication, behavior, and social interaction. Just missed on self-care, and the other ones, show some improvement or even some worsening when it came to sleep or seizures, but these were in 1 individual each. So caregiver assessed quality of life by the Quality of Life Disability Inventory. 14 out of 18 patients showed improvement. On the left-hand side, you'll see the bar chart again, and improvement is upwards from 0, which is along the x-axis in the middle there. And the majority of patients did show improvement. There were 4 patients that were judged to be having a lower quality of life. And when we looked at the specific domains, improvement in this plot on the right-hand side of the screen shows that it moves to the right instead of moving to the left. And we did have statistical significance on feelings and emotions. So 2 of the endpoints that we came close on but did not reach significance included the ORCA and the MacArthur-Bates. These are both vocabulary and communication tests. The majority of patients showed some trend towards improvement on both scales, but neither reached significance. And then just a few things as part of the caregiver impression of change and the clinician's global impression of improvement. The raters were asked to write down what was behind their scoring? What were the things that they saw that moved to justify the rating that they delivered? And so the things that we heard from clinicians included marked improvement in expressive language, moderate improvement in socialization, teachers noticing improvement in learning, the ability to focus better in school, improvement again in expressive communication, more complex, actually understanding kind of and inquiring about the mother's feelings, and also in individuals with more impairment, more babbling, much more than at baseline, and new phrases, and improvement in gross motor skills, including being able to climb ladders. I assume that's on a slide and not to clean the windows, or coming down the stairs independently. Similar things were noted by caregivers, including using more words, retaining eye contact, less scripting and stimming, safer at the bus stop, more focused and engaged and aware of their environment, people, happier, not throwing self to ground when can't get their way, more attentive, easier learning, attention span great, focuses long enough to complete tasks and try new things, and can now run instead of walking fast. So in summary, NNZ-2591 was safe and well tolerated in children and adolescents with PMS. There was significant and meaningful improvement were seen by both by clinicians and caregivers across 10 of the 14 efficacy measures, and improvements in PMS symptoms occurred in those that are important to the caregivers, including communication, behavior, and social interaction. Next steps for Neuren include we have a planned meeting with the FDA coming up before the end of the year in September. And we will do everything possible to get our clinical trials open and underway mid-year, sometime in the first half of 2025. And in summary, I want to thank everyone for being here. Most importantly, we do have to give thanks and a shout out to our investigators, our clinical trial sites, and most importantly, the children and families that participated in our phase 2 trial. We can't do this alone, and we're thrilled to partner with you. And with that, I'm going to be prepared to answer some questions.
Thank you, Liza. And also, I'd like to apologize to any speakers if I switch back and forth between Dr., your last name, and your first name. I sometimes forget.
Liza.
Yes. So we do have some questions already in the poll, but if we could pull the poll up for Neuren Pharmaceuticals and folks can scan the QR code. Don't forget to like the top questions. But I think first, I was noticing a lot of people trying to capture some of the results while you were presenting. So where can they find those results in the future if they're looking for them from phase 2?
Sure. So these results are actually incorporated into an investor deck that is on our Neuren website. So if you go under Neuren website, and I believe it's under the communications tab, you will be able to find a slide deck from December of 2023 that will include these. And also, many of these similar presentations were available. We did a presentation for Cure Shank earlier this year. I think it was in March, and I believe the slides are there, or they pointed to our website. If you have trouble finding it, just reach out.
Thank you so much. Another question that we had was, I'll start with the top one here. Do you know if this drug helps with incontinence, or how did you measure GI issues? Can we add the ADL of potty trained with this drug? or ADL stands for what again? Activities of daily living. So anything about incontinence or GI?
We did have some improvements in the GI health questionnaire, we don't look specifically at potty training in any of our endpoints. I do believe that there was one participant who became much more efficient at, kind of doffing and donning their trousers after using the bathroom, but we didn't look at that specifically.
And I think it might be difficult with some of these questions because you don't have the data sitting in front of you. So just generally, another top question is, if you're looking at an improvement in communication, does this mean going from nonverbal to total speech or adding a few words? What's been your impression with that?
Well, again, relatively small trial, large age range, and more important than that, we know that, Phelan-McDermid syndrome is a spectrum, so we're really measuring each child from where they are. So the improvements are really rated from where they're starting to their next developmental task. So therefore, that's a kind of hard one to generalize because we don't have a minimum or a maximum as far as what kind of communication skills you need to enter the trial.
The next top question has to do with inclusion criteria, I think, and I know you probably can't list those all out right now because you haven't met with the FDA yet, but generally, do you have any comments about who phase 3 will be open to or where people can find that once you know?
We do have a website. Right now, it's almost a placeholder website, but there is some of the data from the phase 2 trial on it. And the website is, you can either go out to our little table just outside the meeting room and scan the QR code on the table stand, or you can take a card that has the web address to the website. So I would say stay tuned there for sure. And we're always in contact with our friends at PMSF, so they will know as soon as we know, honestly. So we will keep everybody informed. We learned a lot from this phase 2 trial. We anticipate that we'll be able to streamline some of the inclusion and, more importantly, the exclusion criteria. But we need to land on a final protocol before making any commitments.
Next question is, were there any differences that you saw in efficacy generally between older and younger participants?
We have done a light touch analysis on that, and we have more data coming, obviously. But at this point, we don't think within the age range we studied that there was too much of a difference. We saw improvements in both. Again, it really, I think, is so influenced by the heterogeneity of the disease.
Is the drug intended to be taken continuously to maintain its effect?
Yes.
And what form is that in?
Excuse me?
Is it pill, liquid?
Oh, sure. So for the phase 2 trial, the formulation was an oral liquid. It was strawberry flavored. We had no complaints or issues with dosing, but we've moved it to a fruit flavor that is just a little more, well, to be honest, the strawberry, we had natural strawberry flavor in it, so children had to be excluded that were allergic to strawberries, and it was one of the reasons for exclusion. So we've now no longer use real strawberries in the flavor.
Thank you. We have 3 minutes left of questions before we transition. Will next trials include adults, do you think?
And unfortunately, I don't believe so. I think that what we're focusing on, because of the impact we have on CNS plasticity, and as Dr. Barry Kravis reported, that the opportunity to intervene in younger children probably gives us a better opportunity to see an effect. Also, though, we are committed to making the drug available ultimately to everybody. We just don't want we're a little farther ahead on the littles, and we don't think it's fair to make them wait. So we'll start our pediatric trial. We will get adolescent data as those individuals roll over into the open label extension. And so we are hoping to gather initial safety data and open label efficacy data in adolescents in the open label extension trial.
Thank you. And you might have just answered this, and I was reading other questions, but there's a question of, will phase 3 be double blind with a placebo?
Yes, it will. And that is part of the reason why we are committing to an open label extension, to really, put people on parity. If you were assigned to placebo, you will have the opportunity to have the drug in the open label extension period. The other thing I want to add, and is that for the children who participated in the phase 2 trial, if they are interested, they will be allowed also to enroll in the open label extension.
Thank you. I love this question. It says, what exactly does clinical significance mean? How big of improvement indicates, I love this because I think it's a great question.
Wow.
Indicates clinical significance.
Right, so clinical significance is a statistical test, and it's really telling you that your findings are not due to just the randomness in any population or study. I think that's the best way to explain it. And the clinical significance could be on a very small change, or it could be on a monumental change. That's not what significance is measuring. Significance is really measuring, is your result the result you're looking, is it an honest and true result, or is it due to just sort of the randomness of the population?
Thank you. And I love that question because we try to clarify that too, that families should look at what the scale is and what the number improvement is that was seen to see if that's meaningful for them. It might be statistically significant without impacting the family, but it can also. So thank you so much, Dr. Squires. This was great, and we will transition to Dr. Dan Gallo.
Great. Any other questions? Please feel free to catch me. I'll be here through Saturday.
Good afternoon, everybody. I'm Dan Gallo with Jaguar Gene Therapy, and I lead our clinical strategy and medical affairs. Appreciate the opportunity to be here with you all today. Thank you to PMSF and Kate and everybody for the invitation. We're going to tell you a little bit about Jaguar Gene Therapy and then also talk about the phase 1 clinical trial that we are right now preparing for with the intention of enrolling our first patient in Q1 of 2025. So a little bit of background on Jaguar Gene Therapy and who we are. Jaguar was founded in 2019 with the mission of accelerating breakthroughs in gene therapy for patients living with severe genetic diseases. Our leadership founded Jaguar with the intent of accelerating treatments and building on the experience that much of the leadership acquired as part of another gene therapy company by the name of Avexis. So some years ago, Avexis developed a gene therapy for spinal muscular atrophy. That was an AAV gene therapy, and that was ultimately approved for the treatment of spinal muscular atrophy. And so they wanted to take that experience, build on that expertise to ultimately shorten the time it takes to get medicines from the preclinical space and investigational space and ultimately into the clinic. We currently have 3 pipeline programs. These are all AAV9-based gene therapies. The one that's furthest along is JAG201, and this is for disorders where there's a SHANK3 mutation or deletion. JAG201 is currently in preclinical development, and this is for type 1 galactosemia. This is a inherited metabolic disorder. And then JAG301 is a little bit earlier, but also in preclinical development, and this is for type 1 diabetes. And of course, we're here today to talk about JAG201 for Phelan-McDermid syndrome. And so Dr. Lisbury-Kravis talked about a number of different approaches that you can take to gene therapy. In our case, we're focused on gene replacement therapy, where ultimately the goal is to give a treatment that introduces a functional copy of a gene to make up for either a defective gene or a missing gene, but ultimately to restore the function of that gene so that you can get increased protein production in the target cells of interest and ultimately change the underlying pathophysiology, the underlying biology that gives rise to the disease. In our case with JAG201, we're delivering a functional copy of SHANK3. This is known as a mini gene because it's a smaller version of the whole SHANK3 gene. And we're delivering it directly to affected cells in the brain, in the central nervous system, which are neurons, with the goal of increasing the production of SHANK3 protein. The idea there is if you can increase the production of the SHANK3 protein, you can address the underlying issue or deficit in the biology that prevents neurons from creating and maintaining connections. And if you can do that, then potentially you could start to change the course of the disease. This is a therapeutic approach that's been investigated in multiple treatments, multiple diseases. As I mentioned, there are already some approved AAV gene therapies, and there are additional ones in clinical trials. A little bit of background, and Dr. Lisbury-Kravis spoke to this a little bit, but what we're ultimately using are what are called viral vectors. In our case, it's the adeno-associated viral vector, and viral vectors take components of viruses, but they're not functional viruses in and of themselves, right? So viruses are generally associated potentially with disease. They exist independently of living organisms, but require living organisms to replicate. They use the host machinery to create copies of themselves, and depending on the cells that they're using to create copies of themselves, they can cause disease. In the case of viral vectors, what we're doing is we're stripping away any potentially disease-causing components, so any components that allow the virus to replicate, and we're ultimately using the delivery aspects of the virus. So the outside of the virus is called a capsid. It's a protein shell. That's ultimately what's usually required for interacting with cells, providing access to the inside of the target cell. And this is ultimately the main piece of the viruses that we're using. In our case, we're taking that capsid, we are inserting our gene of interest, so SHANK3 mini gene, and we're just using that capsid as a delivery vehicle so that we can get our gene of interest into the target cell of interest, so in our case, the neuron. Once inside, that delivery vehicle goes to the nucleus where genes are expressed, and it delivers our target gene, so SHANK3 mini gene in our case, and it exists in the nucleus as a separate component from the host DNA. So depending on the type of viral vector you're talking about, that DNA that you're delivering may integrate into the chromosomes of the host DNA and become fused in a part of host DNA. In our case, usually what happens is it exists as a separate component, what's called an episome, and it's able to express that gene, turn it into RNA, and then ultimately use that RNA to create more protein within the cell. And so we chose this, this platform for delivering gene therapy for a number of reasons. Some of them I've already spoken to, but ultimately AAV viral vectors are efficient at delivering genes of interest to target cells. And so that's helpful to us, right? Our target cells are the neurons. They're generally non-disease causing, right? And so that's also a positive. They deliver the gene in a way that allows it to exist separate from the host chromosome, and that has benefits. If you're integrating into the host chromosome, there's potential for interrupting important sequences of DNA, which can lead to associated issues, and so we want to avoid that. And ideally, because we're targeting the neurons, which are post-mitotic cells, There's evidence that maybe there's some division in neurons, but ideally, we're delivering it at a point where most of the growth that's taking place in the brain has already occurred. And so that gene will not be diluted out as neurons divide, because for the most part, they won't divide. So potentially with a single one-time administration, you may have a lifetime of benefit. There's also, because there have been so many programs that have come before that leverage the AAV system, there's a history of understanding. There's a database of knowledge of the types of experiments you need to do to demonstrate efficacy, demonstrate benefit in preclinical models, to understand safety, ask important questions about toxicity. We have an understanding of what potential toxicity looks like that may be associated with AAV gene therapies. And so we have the opportunity in preclinical studies to thoroughly evaluate all these things, so that by the time we get to human clinical trials, we already have some understanding of the potential for benefit, but also the potential for risk, right? And if something's too risky, then you don't move forward in clinical trials. If there are things that emerge as signals, you could potentially adapt the way that you're studying in humans. And so Dr. Lisbury-Kravis talked to us about this drug development process that we have up on the screen. This is the general approach to developing medicines, and the general idea being you start with many exploratory compounds early in development. Few of those make it into preclinical development, where you're testing them in animals and potentially animal models. Excuse me. And fewer still make it into clinical trials. First phase is really ultimately looking at safety and potentially looking at different doses. In phase 2, you may start to look at additional doses, larger population, potentially signals of efficacy or benefit. And then phase 3 is ultimately what's required for approval, and that's really where you try to establish the efficacy or the benefit as well as the safety profile. You then submit an application to, in our case here in the United States, the FDA, where it's reviewed and either you receive an approval or you don't. Now, this is the general approach that's taken, and as Dr. Lisbury-Kravis mentioned, in rare diseases, there are a lot of additional challenges, right? There are potentially fewer patients, there's potentially less that's known about disease, and the potential for benefit of different types of treatments, and so we try to combine these phases, and really the takeaway is, as soon as we're going into clinical trials in humans, we're really using those opportunities to learn as much as we can about the benefit or efficacy profile, as well as the safety profile of a treatment. And the goal is to try to, in as short a period of time as possible, effectively assess the safety and the benefit so that you can get closer to addressing the unmet need in the population. So just a few updates. We've done a couple of webinars over the last few months, and really over the last couple of years, we've been focused on our preclinical development stages, so completing all of the experiments that we need to do in different types of animal models, so in rodents, in some of the models that were discussed earlier today, in non-human primates, so really to further evaluate safety and toxicity in non-human primates, which are more closely related to humans, and then ultimately submitting that package of information to the FDA so that we could get the agreement to move into clinical studies. But a little bit of background, as has been mentioned earlier, JAG201 is an investigational treatment for the treatment of SHANK3 haploinsufficiency. And this is a gene therapy where, again, we're aiming to deliver the SHANK3 minigene using the AAV9 vector directly into neurons in the brain to address the underlying SHANK3 haploinsufficiency. The goal is to increase SHANK3 protein expression and then restore the defect in the ability of neurons to communicate with one another. It is designed to be a one-time treatment, and as Dr. Guoping Feng mentioned, this is a program that was developed by him in his lab and our collaborators at the Broad Institute of MIT and Harvard. So a few recent updates. In May of 2024, we did have the opportunity to present some of those data that I alluded to, which we submitted to the FDA, really establishing the safety and efficacy profile in mouse models of SHANK3. These were the data that were submitted to the FDA in June 2024. We had a Type C meeting with the FDA where there was agreement that those data confirmed what we call prospect of direct benefit. This is the threshold that the FDA has for allowing a experimental investigational treatment to proceed into a pediatric population. Again, this was based on the comprehensive preclinical safety and efficacy study, data that we have generated in our preclinical studies. This allows us now to proceed with our first-in-human study, beginning in patients 2 years of age or older, up to 9 years, and with the option as well to expand into adults, so patients aged 18 and older. In July of 2024, we did announce receipt of rare pediatric designation and fast track designation. Both of these are, really ultimately recognition by the FDA that there's a high unmet need in these populations and gives us more access to the FDA, more frequent opportunities for engagement, and hopefully shortens the time to development of our treatment. Our next steps are to complete the updates to our protocol, because as we announced in January of this year, we had agreement with the FDA to begin a study in individuals 18 and older. And so now we're integrating the addition of the pediatric population into this same first-in-human study. We do intend to complete this likely over the coming months. We're already actively engaged with our clinical trial sites and are fairly well progressed in what we call study site activation. And again, our goal is to dose our first pediatric patient in Q1 of 2025. And so I think, much of this has been spoken to earlier today, but, ultimately SHANK3 functions as an anchor. It plays an important role within the neuron to anchor what we call receptors. Those receptors are important for communication between neurons. The way I think about it is when you don't have enough SHANK3, your ability to create connections between neurons and facilitate communication is impaired. But the ability to maintain those connections is also impaired. And those connections are ultimately what's fundamental to being able to create memories, to learn, to interpret things that you're seeing and experiencing in the environment. And so our goal is to deliver JAG201 to increase the SHANK3 expression so that now there's more SHANK3 in the environment in the neuron at the synapse is more conducive to building connections between neurons, but then also to sustaining and maintaining those connections. And that becomes the foundation for learning ultimately. And this is what we're aiming to do with JAG201. So some details on our study. This is a first-in-human study. It will be open label, which means that every participant in the study will have access to treatment. The key objective is to evaluate the safety of JAG201. So initially at one dose, and then in a second group of individuals at a higher dose. So safety and tolerability. But again, because this is a rare disease, we really want to take every opportunity we can to understand as much about the potential benefit of the treatment as possible. And so we have a robust list of clinical outcomes where we'll be evaluating for potential benefit, many aligning with the presentation we just saw by Neuren. And really what we've tried to do with our study is, as much as possible, build on the knowledge that we've gained from the Developmental Synaptopathies Consortium to understand where are the deficits and what types of deficits can we measure, what are potential early markers of benefit that can give us an indication that we are changing the biology, understanding that some of these longer-term goals, improvements in cognition, improvements in adaptive behavior, not only are they going to take more time, but they're going to take more experience, right? So the individual being able to benefit from that improved biology that is more conducive to learning, but then the experience is to actually go out and learn so that it's translated into a benefit in cognition or adaptive behavior. So we will begin our first-in-human study in young pediatric patients, as I mentioned, individuals in the age range of 2 to 9. And the goal here, the idea here is, as Dr. Lisbury-Kravis mentioned, the potential for benefit may be greater in a younger individual, an individual who is still actively undergoing neurodevelopment, The idea would be to deliver the treatment early as the individual is developing, correct the underlying deficit in the biology, and ideally put them on a trajectory that's more representative of an unaffected neuronal biology. And we aim to start in this population because if we can demonstrate a benefit here, it opens up the opportunity to expand what types of populations we're evaluating. If we were, for instance, to start in an older population and the benefit was not as obvious or decreased, it may decrease the potential for us to then go low, into a lower age range, hoping to see a benefit. And so that's the idea here. But the goal is to start young, demonstrate the benefit in a limited population, and then start to expand on that population as we move further into clinical development. We are finalizing our protocol. Once we are at that point, we intend to make it available via clinicaltrials.gov. Of course, we'll work with PMSF, we'll work with CureShank, we'll make sure that this information is available to you all as soon as that does happen. We've received a number of questions as far as more specifics on that population, and there's some things we've shared to date. We intend to share more again as we finalize that protocol, but a few things I can share today. We'll be starting with a small group of individuals, right? Again, the focus is safety. We have never tested JAG201 in a patient population, so we first want to make sure that it is safe. Pediatric patients ages 2 to 9. We will be focused on small deletions as well as loss of function mutations. And in terms of what those deletions look like, this really gets back to, again, the learnings that we've been able to take from the Developmental Synaptopathies Consortium Natural History, where they divide deletions into class 1 or variants, and then class 2 types of deletions. So class 1 is essentially SHANK3, and then on one side, the 2 adjacent genes, and on the other side, the next adjacent gene. So deletions as small as 4 individual genes. Class 2 is anything larger than that. We'll be focused on class 1 and then loss of function mutations. The idea here is, we understand that we're delivering SHANK3, and that's the piece that we hope to correct. And so the closer we can get to identifying a population, again, initially in these first few individuals, where the only thing that's affected is SHANK3, it just gives us a cleaner opportunity to evaluate the benefit. But that doesn't mean in the future we won't look at other populations. To this point, the inclusion criteria will, the goal will be to, again, identify a population that's a little bit more homogeneous so that we can evaluate that benefit. And then ultimately the goal is to evaluate JAG201 in adults after we've had the chance to evaluate it in pediatric individuals. I've mentioned this, but our goal here is safety and then look at disease response. We're going to be testing 2 individual doses. We've also received questions on dosing, and with the doses, this is ultimately informed by our preclinical studies. So at what doses in our animal models do we see benefit? And then how do we translate that based on the brain biology, the brain size, to non-human primates, to humans? And how do we identify what we feel confident is an effective dose? And on the other end, based on the toxicity studies that we've done, where we test at much higher doses than that, what's a reasonable dose to escalate to that still gives us coverage from a safety perspective, so that ultimately we can provide an increased dose, but still one where we have confidence that our safety profile in non-human primates suggests that it would be appropriate to evaluate in humans. Our initial study will be limited to a few sites, and we've mentioned these before, but we do intend to dose our first patients at Rush in Mount Sinai, so Rush University in Chicago, Mount Sinai in New York. We hope to expand to that, to other sites that are currently involved in the DSC, so potentially Boston Children's, but other sites in the DSC as well. The protocol, as I mentioned, we hope to finalize this here in the next few months, treat our first patient in the first quarter of next year, and hoping for a positive outcome, which would allow us to expand the population of individuals that we evaluate JAG201 in. As far as administration, Dr. Lisbury-Kreva spoke to this approach, but we aim to deliver JAG201 via ICV injection. And she spoke to the procedure here, but ultimately a small hole, what's called a burr hole, is made within the skull to give access to the catheter that is pushed through the brain into the ventricle. That ventricle you can see there is the brownish space that sits in the center of the brain, where ultimately there's CSF or cerebrospinal fluid. We would extract a volume of CSF to allow for delivery of the volume of the gene therapy. And the idea here is, by diffusion, ultimately giving access to the cells of the neuron. It will be administered, the neurons in the brain. It will be administered while the patient is under anesthesia. And, based on the number of ICV administrations that are done, as Dr. Barry Kravis mentioned, across a number of diseases for a number of conditions, we have ultimately selected this one and have confidence in the overall safety profile of ICV administration. And so what's next? Again, we're looking to finalize our protocol here in the next couple of months and ultimately complete clinical trial site activation, allowing us to dose both pediatric individuals as well as adults. And the goal here is to enroll our first patient in Q1 of next year. We have, as I mentioned, early markers of benefit that we would look to evaluate. So, ideally things that may change in, the range of, months as opposed to years. And then we'll continue to follow these individuals for a number of years to understand what is the long-term benefit and safety profile of JAG201. So with that, just want to say thank you to PMSF, to all our collaborators within the DSC, to CureShank, to Kate for this invitation today, and for, again, all of the families that contribute so much of themselves and their experience to allowing us to continue to move this field forward. So thank you very much. I'll take any questions.
Thank you, Dr. Gallo, and you all know the drill with the Q&A. Please scan the code. Please upvote your favorite questions. One of the questions I saw earlier that I think is one we get a lot is, can you speak at all to, the question was how many gene therapies have passed FDA approval? I don't know if you know the exact number, but can you say anything about previous approvals and if there have been any?
So there certainly have been a number of approvals, and approvals for gene therapies, including AAV gene therapies, administered via different routes of administration. Right, so I believe the very first one may have been an ocular delivery one. Zolgensma is an AAV delivered via IV administration. And there have been additional ones, including non-AAV. I actually just, I think I took a snapshot recently of the number of gene therapies that have been approved, but I don't remember the number off the top of my head. Yeah.
The next highest question is, will the age 10 to 17 population be allowed to test at some point?
Yeah, so that's the goal, right? I think, initially we want to understand, can we demonstrate a benefit, right? And if the young pediatric population gives us the best opportunity of demonstrating that benefit, that gives us the confidence to understand, well, what does that benefit profile look in different populations, including older individuals, right? Including up to, as we mentioned, older than 18. But additionally, different profiles, right? So instead of just small deletions and variants, also larger deletions, understanding that the range of benefit may be different, but again, ultimately, if there is a benefit, we would aim to understand what that looks like.
Are you able to answer what were some of the improvements seen in non-human primates? And Dr. Feng is right next to me if we need him to add into it, but do you have any answers for that?
So specifically in non-human primates, it would be Dr. Feng's work, and maybe just to highlight some of the differences. So the difference between the gene therapy, the investigational gene therapy he's using in the non-human primates versus what we're using is the capsid, as well as the delivery method, the administration route. So the gene sequence that was developed by Dr. Feng in his lab, that piece is the same. And so the SHANK3 and the way that it functions, that gene sequence and the resulting protein is the same. In Dr. Feng's case, he's using a different capsid, which allows him to deliver it via IV administration. And he, spoke to some of the types of behavioral benefits and cognitive improvements, social changes that he saw in his marmosets within the cage studies. And in our case, right, we're using a different capsid that's specific to humans, and we're delivering it directly into the brain. But yeah, so for any questions on Dr. Fang's work, I'd maybe defer those benefits to him to speak to.
Perfect. This is a tough one. When a gene therapy is approved, if it is, will it be available worldwide? Generally, how does that work?
Yeah, happy to speak to it. So our initial program here is focused on development within the U.S., right? And so our regulatory discussions have been with the FDA. And so our agreement, our ability to move into clinical trials is based on that agreement with the FDA, which is specific to the U.S. There would be opportunities to have those same discussions with other health authorities, and that may require running additional studies. In some cases, when you have an approval in the U.S., that potentially opens the pathway to approval, or at least to access in some countries where they'll accept a U.S. approval. In some cases, it may require additional studies. Just given the size of a company that we are and the resources that we have, we're really, hyper-focused on this initial study, demonstrating benefit, and doing it in a way where, we can focus in a single geography. Of course, if there's benefit and we can, find a pathway to approval, we would explore additional options to bring it to different geographies.
This is also a tough question, but they ask, can you mention any trial that is similar, gene therapy or not, that may have had some challenges or went difficultly, or can you speak to anything like that?
Sure. Yeah, I think that's a broad question, and I think Dr. Elizabeth Kravis is probably maybe one of the most knowledgeable individuals on that, just given her exposure to different types of gene therapy programs, different neurodevelopmental disorders. She highlighted a number of the challenges that you can encounter, right, which really range the full spectrum, right? So, in addition to being able to, generate a clinical data profile that speaks to the efficacy, that speaks to the safety, there's a number of hurdles that you have to climb with the health authorities to demonstrate to them that your product truly has a clinical benefit. A clinical benefit that ultimately justifies an approval and a safety profile that supports it. I think in our space, in the neurodevelopmental space, that really tends to be, at least from my perspective, maybe the biggest challenge, right? Selecting the right instrument where you can say, we're seeing a benefit here that, as Liza spoke to, is clinically meaningful, right? So it's a benefit that's interpretable, understandable, and clinicians and families would agree this is meaningful. It's statistically significant, right? So it's not just occurring by chance, but it's a benefit over what you would expect in a placebo population. And how you get to that is a challenge, right? We have these different types of clinical outcome assessment tools, but the FDA's threshold for what they believe is a meaningful benefit is going to be different from, disease to disease, instrument to instrument. I think that's probably the biggest challenge.
If you've had one AAV9 treatment, and this is the delivery system, e.g. JAG201, can you receive other AAV9 treatments, e.g. JAG301, or does the immune response pose a concern for future AAV9 treatment?
So I think if we look at other gene therapy programs, what you'll usually see is an exclusion for any experience, any prior treatment with an investigational gene therapy. And I think it's what's been alluded to already, which is once your immune system sees AAV9, even though it's not infectious, your immune system still perceives it as foreign. And so it's going to attempt to mount an immune response to it, right? Doesn't mean it's infectious, doesn't mean it causes disease, that's how our immune system works. And so if you were to try to administer a subsequent dose several weeks, several months later, the immune system's gonna remember that. And there may be antibodies, other parts of the immune system which recognize that second dose and remove it, eliminate it, thinking that it's infectious. this is ultimately up to the sponsor, right? And so what I mean by that is there are immune privileged sites, right, within the body where the immune system won't readily mount an immune response. Potentially part of the brain would be like that, potentially the eyes, other organs. And so it probably depends on, that individual company developing that gene therapy, how it's going to be administered, where it's going to be administered, what it's treating. So more than likely, if we're looking at other clinical trials right now, prior experience with an investigational AAV9 is excluded, but that's not always the case. Yeah, it really depends on where you want to deliver it and how.
And similar to that, would a participant likely need to be on immunosuppressants for a trial like this?
Yeah, so that's a great question. So for the same reasons, and this is an evolving space, but for the same reasons, there's always an immunosuppression regimen that's administered. And the goal here is to maximize the delivery of the gene therapy, but then also to prevent the immune system from overreacting to seeing the gene therapy, both of which would be a challenge, right? So on the one end, if your immune system overreacts to the gene therapy, that's a safety concern for the individual receiving it. On the other end, if it overreacts to it and prevents it from getting to the target sites, well, that's going to affect the benefit that you see from it. And so there will be an immunosuppression regimen in our clinical study. And ultimately, we design that based on what we see preclinically, but then also the history that's understood from other AAV gene therapies, as well as the fact that we're going into the brain and not delivering it systemically via IV administration, which would result potentially in a larger immune response.
Thank you. And I think this will be the last question. We've already been asking Dr. Gallo 10 to 15 minutes of questions, so we're wrapping up for the day. But the last question is, if a child is in the first phase and starting on a low dose, but throughout the clinical trial a higher dose is found to be more effective, is there a possibility for a second dose to compensate?
So I think those are decisions we haven't made yet. Again, I think if we look traditionally, if you've been administered one dose, generally that will make it an exclusion criteria for a subsequent dose. What I will say is that first dose that we're testing, that is based off of the preclinical data that we've generated. And so we go into our first in-human study at our first dose with a level of confidence that what we saw, the dose that we used in animals that translated into a benefit is comparable, right? And so we're delivering what we have made the case as an effective dose. That second dose, it could be that it's as effective. It could be that it's more effective. Potentially it's less effective, right? That part's less likely, but we don't know. And so it gives us the option to test a second dose if, for instance, that first response wasn't so robust, right? We make the case to, inform the selection of that first dose so we do see a robust response, but that second dose really gives us a second option in case we do not. But those would be decisions that we would make, I think, at a future point.
Thank you very much. Please join me in thanking Dr. Gallo.